Everybody. I'm Andrew Kendall. I represent ESI. Thank you for joining us today. We've got two speakers, Graham Taylor, who's chairman of ESI, and Bruce Whitelaw from the Roslin Institute. The plan is that they will make a short presentation, or it'll be about half an hour in total between the two of them, and then we'll have a discussion and questions. So can I ask you to mute your microphones, please? And then when they've finished, we can then sort of open up and have a discussion. So without further ado, Graham, we'll hand over to you. Thank you.
Lovely, thank you. Yeah, I'll just put my presentation up. Hold on a moment. Screen share. Can you see and can you hear? Brilliant, excellent, thank you. Well, yeah, thank you. Well, welcome to all who've signed in this afternoon. I've been introduced. I've been chairman of ESI for 3 or 4 years now. I'd like to thank you for putting the time aside to attend the webinar today. We had an earlier one with Con4 and CLA, and there was kind of 80 or 90 signed in, which was really helpful. And we're just starting to kind of make more noise about the prospects for gene editing and the options that present to us through doing so. I joined ESI in 2002. Really, I was a young forester at that point, and I was just it was out of sheer frustration at what I felt was the lack of impetus on the really terrible impacts of grey squirrels are having on young woodland. I remember being in the room with some of the great foresters, you know, Miles Barn, Bede Howell, Peter Goodwin. And for myself, it was the irritation of my work being completely and utterly destroyed by the grave pest, despite following best practice with warfarin that was available at the time, despite working hard and trying to get trapping and shooting to get populations down. Despite all of that, despite spending lots of money on getting young trees up and away, along could come year 7, 10, whatever, and your work is destroyed. And that's driven me to support ESI, as well as engage in red squirrel conservation in properties where we have red squirrels as a company, where we are managing those woods. Let's move on. So my day job is running a business called Prior and Rickett Silviculture. We're slowly, well, fast actually, becoming a kind of a national entity. We've got multiple offices around the country. But I particularly spent most of my career in the Welsh borders, on the English side of the Welsh borders, working in the marches. Now, it is one of the best oak growing parts of the country. But sadly, as a result, we also have the greatest propensity for squirrel damage because there's a correlation between fast growth and damage to oak trees. And in fact, it's not just oak trees, there's a whole host of species, because many of the broadies grow so beautifully well in the Herefordshire, on those Herefordshire deep red soils. Damage, as we know, is widespread across many of our beloved tree species. This slide illustrates just the susceptibility of each of these species. And of course, many of our natives are on the left-hand side, at the highly susceptible end. And actually, if you take it coldly, looking across the nation, we're slowly pushing to a forestry world where those trees are a diminishing part of the mature forest, and other species are becoming a more dominant part of the mature forest. So whether it's on the conifer front, Sitka spruce and Douglas fir, very, very happy, nothing's bothering it too much at the moment. And cherry, alder and lime are beginning to dominate in the broadleaf components. It's going to take a long time, but that's the process. And we've seen, during my working life, significant swathes of new planting during my career. I can think of a number of kind of regional impetuses. The Southwest Forest back in the 90s, about 3,000 hectares of woodland was established, publicly funded, an impetus to, yeah, we must get more woodland in the landscape. It was about 2,000 hectares of broadleaf and 1,000 hectares of conifer. And a report just recently, 18 months ago, 2 years ago, by the folk in the Southwest from the Forestry Commission, highlighted that, you know, there wasn't an oak tree of note. They'd all been damaged by the grey squirrel. The ash has been succumbed to cholera. There's a bit of alder, and the only species that are kind of doing well and are healthy funnily enough, the Sitka spruce and Douglas fir that constitute the 1,000 hectares out of the 2. Similar concerns in the national forest. We've been planting a lot of money, a lot of resources going into the national forest. 20 years on, speaking to the National Forest Company and the people that are managing it, it's hard work. They're getting a lot of damage. They're trying to do control with current technologies, and it isn't really working very well. Reports in the 90s from Forest Research, who did some analysis in Wales. This was reporting, and they went and studied private sector restocks. What are they looking like? And they all came back, and over 90% of grant-aided restocking have been utterly trashed by the grey squirrel. And I spent more time locally in southeast England, well, I say across southeast as the business has grown. And I spent time looking at areas that blew down in 1987, were subsequently restocked with a lot of public support, a lot of energy, and the oak, beech, sycamore, sweet chestnut, all badly damaged, except where people are really enacting strong levels of control. So this isn't a minor problem. It's a major problem, and it's a stuck record because it keeps carrying on. So I just thought sometimes it's easy to just go into wood and think, well, that's just like, this is what a normal woodland looks like. So I've got some slides now that just illustrate the impact of bad and/or medium grey squirrel damage due to trees. And it is normal to see it in the UK. You can see it everywhere in UK woodlands. So, and it's public woodlands, private woodlands, woodlands owned by NGOs, woodlands owned by small woodland owners. You see it everywhere. So this particular slide, taken of a stand in the Chilterns. It was private forest in the Chilterns in the blew down in the 1990 storm, replanted with oak and other broadleaves, predominantly oak, and they're literally flat topped. They're not growing up. The whole stand is flat topped, all caused by grey squirrel damage. Stand image on the left there is a forestry mission site in the Wyre Forest. It's a leasehold site. Clause restoration was the agenda, has been the agenda, continues to be the agenda. Spruce were felled in that stand in the late 1990s, oak planted at random spacing, and then nature allowed to take its course. No squirrel control. That's what you get, and it's acres. It's not just the odd tree. This is acres of stand in that condition. Stand on the right is a stand that's in an estate in the East Midlands where we just started getting involved as a company. All the last 20-30 years of restocks are looking like that. Apical dominance, broad branches all over the place, and not really going upwards at all. Next slide. It just shows more of the same, really. This is 500 hectares of private estate in Berkshire. Same story, girdling, poor form. That will never, those trees there, that stand there will never recoup the value that was first put in by the landowner and actually indirectly by the state, which I'll come to in a little while. And success is rare. Part or complete failure is the norm. And the failure to get past that damage phase has a long-term impact on the economics, but also the structure and the biodiversity interest within the forest. So I just thought I'd show you some good examples. It's kind of, this should be normal, but it isn't. So on the left there's a beech tree growing healthily because we zero tolerance to squirrels on the estate, spend lots of money, tens of thousands of pounds, but to keep killing squirrels, keep killing neighbour squirrels because they backfill. And there's the result. Image on the right is a sweet chestnut. It's not quite so clear, that image. Apologise for that. It's in Wales, 15-year-old sweet chestnut in Wales. That's image of 25-year-old oak in Herefordshire. It's not, again, a fuzzy image, I'm afraid, but 15 to 20-year-old oak, beech and sycamore in the Cotswolds, all being controlled with current technologies. Image on the left there, again, North Yorkshire beech growing happily, beautifully upright. And the image on the right is a bit of a pointed one. It's actually not an English oak tree. It's a French oak tree. I had a study touring tour about 3 or 4 years ago. Point being, that is normal in France because they don't have the grey squirrel. What I've just shown you in terms of the damage is now normal and has been for quite a number of cycles in the forest. Total losses. Well, in terms of timber, I did some work using the forestry inventory data, and my best educated guess, we look after quite a lot of hectares of broadleaf forest across the country, different settings, and my finger in the air, desk-based assessment, I reckon was about £40 million a year it's costing the forest owner. Interestingly enough, UK Squirrel Accord and RFS commissioned some work, and they came out with a similar number, so we're probably about track. Point though is that that's annually, and this has been going on probably for 50, 60 years now. So actually there's a cumulative loss, and a cumulative loss that's dripping out of the forestry sector. So that's timber loss, but you then add to that expenditure to try to win the war, where you actually sometimes win and sometimes lose. But this has an impact, it has a drip impact on the forest now. The effect in the future is still to be felt, because we are currently felling oak trees that were planted and managed during the Victorian era, when there were no grey squirrels, or there were very few around. And so those trees grew healthily. From about 60 years' time, we'll start to feel the effects of the last by that stage, 120 years. And my best guess is that that'll be about another £110 million a year on the import bill. That might, say, well, that's a rather large number. We import about £14 billion of the hardwood timber. That isn't a large number. We're just going to add to the import bill, and that will be a drain on just national resources, but also it's a drain on the forestry, on the multiplicity of private sector forestry accounts. Property damage, I've just had some, funny enough, I've had some at home here. I think the squirrels know where I live. My broadband line went down about 3 weeks ago, about 5 weeks ago, for about a period of 3 weeks, and they had to restore a whole load of cables caused by squirrels. But the British Association of Insurers have published a figure of £20 million a year. Carbon losses, haven't yet tried to work that out. There will be an impact. Those trees that I showed you in the Chilterns that aren't going up are not sequestering carbon like they should be. You get different effects with mixtures, and with modest amounts of damage, there's probably a more modest effect, but nevertheless there's effect. And of course, once carbon becomes more heavily priced, that cost will be bigger. Biodiversity. What price do you put on a succession not happening in a semi-natural woodland? What price do you place on being confident to restore pores, having a choice between Douglas fir, which grows at yield class 20, behaves normally, puts on lots of value every year, or squirrel damaged junk? And that's the choice that many private landowners face, and not surprisingly, they're a bit reluctant, to put it mildly. But other impacts on wild birds, mammals, the ecological footprint of the grey squirrel is much heavier than the native red, and I'm convinced it's having an impact on other small mammals in the system. Dormice in particular, I think they are having an impact. So what ESI, what have we been doing about it for the last 20 years? Well, we were supportive of the IMC work. We've helped with trapping technologies. We've got good partnerships with Italy and Ireland, where they've also got grey squirrel problems. We funded work with the GWCT, ongoing PR work, discussions with UK and European politicians, helped develop, pushed along the Alien Invasive Species Directive, and have done various studies on the effects of grey squirrels, published lots of books, supported an establishment of the Red Squirrel Survival Trust, have done quite a bit of research on other things and different control mechanisms. That's all I really want to say, other than we are very excited, having spent 20 years trying to push all these other things along. We think the prospect of gene editing is the thing that could really be a game changer. It could really dramatically shift the balance from this sporadic drip of cost that is across affecting red squirrel conservation spending and owners who are trying to protect their trees. I haven't done that number, but it's quite a big number. Much of it's actually investment by government indirectly. Making that shift towards actually seeing the reds begin to become re-established in more widely across the country, but also our forests behaving normally as opposed to abnormally. So we're quite excited. I'm going to hand over to Bruce now to explain the science, because he's the scientist. I'm just a modest forester, really. Bruce.
Thanks, Graham. I'll share my slides. Are you seeing this?
Yes.
Okay, so for those listening who have not met me before, my name is Bruce Whitelaw. I hold the chair of animal biotechnology at Edinburgh University, and I'm currently the interim director of the Roslin Institute. I've been working with genetic engineering technologies since I graduated as an undergraduate. In fact, it was in the last year of my undergraduate that I got excited about the idea of being able to manipulate genes in animals and in organisms. Through that time, tools have come and gone with the trendiness of them, and you will all be well aware that the current exciting technology on the block is CRISPR technology or gene editors, and we are using that. To put in context, we picture of beautiful Edinburgh with blue sky. That wasn't taken today, I should add. Roslin Institute is based just outside of Edinburgh on the Easterbush campus. It's in effect, it's the animal campus of the university. We are right next to the vet school, the Royal Dick School of Veterinary Studies. And I'm currently sitting in our innovation center right next to it. What is Roslin famous for? Well, a certain sheep, which we produced 25 years ago. We're just going through throes of celebrating what would be Dolly's 25th anniversary. What have we done since? Well, we've kept on going. In fact, one of the drivers for making Dolly in the first place was to help in increasing the efficiency and accuracy of genetic engineering. And as I indicated, there are now really very powerful tools with huge potential called CRISPR. Apologies for that typo there. CRISPR-Cas9 being one of them. There's a range of genome editors. There's base editors, talons, zinc fingers, and so forth. And we work with all of these in various projects, trying to see how they can improve the welfare and health of animals. And our lead project, which I'm sure many of you will be aware of, revolves around pigs. It's a project we're doing with genus PIC, and it addresses a viral infection of pigs called PRRS, porcine reproductive respiratory syndrome virus. We have identified, we've used candidate gene known to modulate the ability of that virus to get into cells in culture in vitro, and we've edited in animals. We removed a single exon using 2 guides in the CRISPR technology, removed domain 5, and these pigs are resistant to infection. And genus PIC are currently working with the FDA and USDA in the States to bring them through the regulatory process. This is my day job. I lead projects around engineering animals. We're currently working on, in my own group, cattle, sheep, pigs, and we're looking to goats across the institute. We also bring in poultry, chickens, and we're doing the same in some of our aqua species that we're interested in, particularly the salmonids, but we're looking to do the same in tilapia. This is the research team I brought together for the project Digby. I'll explain what that is in a minute. It's a range of academics, plus our business development exec from Shan, from Edinburgh Innovations. And across this team, we've got expertise in the genetics, the molecular biology required, engineering of animals, large and small, but also modeling, and I'll come to that in a few minutes. We proposed a few years ago in a prospective article that we could use CRISPR-based gene drives for pest control, identifying squirrel as one of the potential candidates. At that point, it was theoretical that gene drive would work within mammals. It's a technology that's been developed in insects, and I'll come to that in a minute too. But since our paper, it's been shown to work within mice. A group in the States have done this, and recently the Australian government has put money into gene drive strategies for controlling the mice rodent plague that island is suffering from. And just to draw your attention to an interesting aspect of this money that's gone into gene drive control in Australia, it is endorsed by PETA. an organization not known for its support of biotechnologies and livestock. So what is DIGBY? DIGBY is direct inheritance of gender bias. And again, I'll come to that as I go through the presentation. But first of all, what is gene drive? So, and I apologize for those of you who are knowledgeable in genetics. This is a very simplistic diagram here, but it gets the message across. So as you know, in sexual reproduction, boy meets girl and offspring are produced. We all carry two copies of a gene, two alleles of the gene. And in all cells in our body, apart from our gametes, whether that's sperm or egg, which carry one copy. So a father can give copy A or B, and mother can give copy C or D to offspring. What that means is if there's a gene of interest, a target, a genetic loci of interest, it will be passed down to half the offspring, whether it comes from mum or dad. And unless there's selection pressure applied to that, it will stay at that low frequency through the population. In the case of gene drive, the offspring that are produced, or the gametes that are produced, all carry the gene drive transgene, and it copies itself immediately, and I'll explain that in a minute, in such a way that over a short period of time, it drives it through the population to theoretically 100% of offspring. So the top part of this slide sort of illustrates how the gene drive works. We insert the transgene into one of the alleles experimentally in an embryo in a lab environment. And that transgene encodes for the CRISPR-Cas9 tools that are then activated. They then engineer the copying of the transgene into the allele in such a way that all animals are homozygous. So if you've got a male who's gene drive, it doesn't matter if he passes a Y or X chromosome, along with it will go one copy of the gene drive. Once the sperm fuses with the egg, that gene drive is then copied into the other allele, and so on and so forth through the population, driving that presence of the gene drive over generations. Grey squirrels, well, I can't remember exactly when it was, but I do remember where it was. It was about 50 feet from where I'm sitting just now. I met up with Andrew and colleagues from ESI, and we started discussing a genetic approach to controlling grey squirrel numbers. And the idea here is that we would drive fertility through those animals. Now, gene drive could be used to drive any trait through a target population. If you go back to the PRRS pigs, if it was deemed financially attractive or commercially or industry attractive, we could structure a gene drive that would drive, potentially structure a gene drive that would drive PRRS resistance through the population. You could drive any trait. We are looking at disease resistance in the context of the grey squirrel as a population control. We could drive female or male infertility. It's quicker to go with female infertility because the boys are more promiscuous than females, but you could do either way around. We've come a little bit with the initial support from ESI, funding support from ESI, allowing us to do some modeling and create some of the initial sort of test constructs, reagents for that. I'll come on to that in a minute. At this point, what's really attractive to me is that gene drive offers, in comparison to the current control mechanisms within the greys, or indeed any pest species, a humane, no kill, no trapping, no poisoning solution to control their numbers. So conceptually, to illustrate how we would achieve infertility, I've got two examples here. One is called, quite excitingly, the X shredder. And in this case, the gene drive's task is to destroy the X chromosome. So therefore, any gametes produced do not have an X chromosome. It has challenges attached to it in that you need to have a very precise activity of the gene drive. The homing strategy is conceptually simpler. You identify a gene involved in fertility, in this case female fertility, and you target the gene drive to that gene, it copies itself into the other allele and thereby inactivates that gene, leading to infertility. In this way, we would drive it through the population. The reason why I've got insects in the picture here on the right is this is where most of the activity with gene drive is happening today. There's a lot of interest around using gene drive to control mosquito populations. We are already seeing genetically engineered mosquitoes being released in other parts of the world as a sort of forerunner to this, but the gene drive would take that to a more effective approach. And I strongly, there's a very big difference between insects and mammals. Insects reproduce much quicker. The progress that colleagues will make within the insects community around design of gene drives and implementation and the hurdles to cross, I think we can learn for anything we want to deploy in pest control here in the UK. What do we do with the support from ESI? As I mentioned, we made some initial constructs and we validated those in some squirrel cells that we generated. And so they work, they'll target sequences and so forth, and that's a great start. But we also modeled the impact that we could see with deploying gene drive. This is a very simple model study with just very few parameters looked at. We looked at two population size, 3,000 and 30,000. We looked at randomly mated and specific deployment of the gene drive, releasing 100 gene drive squirrels with an estimated 12-month breeding cycle. And as you can see, depending on the size of the initial population, 100 gene drive animals would lead to a reduction in that target population over a 2-decade period. Halfway through the first decade, you would see a significant reduction in the number of animals with approaching eradication after the second decade. We obviously need to do an awful lot more modeling to fully understand how best to deploy such animals, but this is an indicator of the sort of timescales that we might expect. It's a platform technology. I mentioned that we could use this in insects. Well, we could use it on a whole variety of pest species. We've had discussions around rodents, not with groups here in the UK, but with groups outside of the UK. We've had discussions around crustacea like the signal crayfish, but really any animal that sexually reproduces and doesn't have too long a reproductive cycle. If you were going to target an animal that took 20 years to breed, then gene drive would be a very long process indeed. Right, just to summarize before I pass you back to Graham, the proposal here is to use the new genetic engineering tools, genome editors, transgenic technology to create a gene drive which would drive infertility through the target population, in this case, grey squirrels. There are ways to build in stops, which have been shown to work within insects, and the daisy chain is one example, and I can talk you through that if you want. But really, there needs a lot more work to be done in that area, and as I said, I'm anticipating that emerging soon from the insect community. We already have trials going on and significant progress in immunocontraception strategies for controlling greys. This is really just the genetic equivalent of it, and it's not an it's not an or type proposal we've got here. Gene drive can work quite well with immunocontraception, and I'm very keen to sort of model that type of, as with other controls mechanisms, I'm quite keen to model the sort of dual impact that these things might have, with immunocontraception coming in early and gene drive being used to sort of lock the effect into the target population. As I said, it can be applicable to a range of pest species, and anything that we learn from developing with grey squirrels will have direct application into other species. I emphasize again, it's humane, no animals are killed. I'll also emphasize it's specific for the grey squirrel. There's no collateral animals affected by it, and this is absolute. The gene drive functions by recognizing a specific sequence. We have sufficient, more than sufficient genomic sequence data to design it such that it will only function in a grey. In fact, we could go beyond that to what people are calling private sequences. If there were subpopulations of greys that had a specific sequence, it's conceivable we could design a gene drive just for that subpopulation. As an example of that, there are regions of the UK that have greys which are cut off from other grey populations, Aberdeen being an example of a sort of an island of grey animals. How long? Well, I'm estimating it's a 5-year development program. There's some biology in there that we need to overcome and address, and that makes it not absolute tight that it'd be 5 years, but that's what I'm anticipating, and I think we might be able to do it quicker than that. And then I think you'd have a 3-year trial, contained use, limited trial phase before it could actually be deployed. And then if you did deploy it, you'd then start to see an impact in the second decade. And at that, I'm going to stop sharing and pass you back to Graham.
Hold on. I'm just trying to get my share screen again. There we go. Okay, can you see that again? It's not working for some reason.
This is on my screen, Graham.
It is. Oh, right. Okay. I'm just trying to get it. Hold on a minute. Let's go back. Can you? Oh, okay. Screen sharing has stopped. Go back to screen sharing. Share screen. There we go. Right. Be with you in a moment. Share screen.
Key thing is to press the slideshow thing on your active screen and not on the thing that you're sharing.
Yes, yes. There we go. I'm back to where I was. And I need to change my display settings, swap presenter view. Apologies. I'll just scroll through to my, it was just a last point really. I was sitting thinking last night about my career. What's happened in specifically English forestry, because it is what's mainly happened because of government grant support and the push for broadleaves, is that mainly we've been planting broadleaves in England over the last 30 years. And I turned to the stats that published very usefully by the Forestry Commission every year. And during the last 30 years, Forestry Commission England have felled and restocked, well, they've restocked with nearly 12,000 hectares of broadleaves, which is about 400 hectares a year. Much more significant amount of felling in private sector. I mean, there's more significant private sector land holdings in England. And typically that's been 1,200 hectares a year of private sector restocking. One thing that I was encouraged by in one sense is that I think in my career last year, we pushed through 100,000 hectares over that career, 30 years, which is quite an achievement in some ways. It's the last few years, I know we've been failing woefully short of government ambition under the Environment Plan, but nevertheless, we've planted over 100,000 hectares of new broadleaf woodland in England in the last 30 years. I then sat and thought, well, how much does this cost? And I used, these are average figures because it's very difficult to tell, because at any one time there's a whole series of supplements, whole series of hectarage payments that vary depending on the extent that you're planting. Farm woodland scheme rates varied by land type, etc. But I just used average figures at the time, which I thought was a fair assumption. This doesn't include the supplements, and this doesn't include the administrative costs of administering. These are the direct payments that DEFRA and the Forestry Commission have made to private landowners. The costs for FE restocking, I've assumed the grant level, because it would have cost the Forestry Commission something, but I've assumed the grant level. And that came up with I came up with a quite interesting figure there. It's £252 million for the planting grant, and about 430 odd million pounds for the attached farm woodland scheme and farm woodland premium scheme payments. That's very close to the 650 million pound Nature for Climate Fund that we're just about to unleash, planting more woodland. That's going to cost more. I think the average grant rate will be now closer to about £10,000 a hectare. So if the target is 7,200 hectares a year, that's £72 million, rising to 10,000 hectares a year, which is the ambition. We'll be talking about £100 million a year, public money going into planting trees. I know, looking at my career, just looking at the woods that I've been involved with over my career, that despite all the best technologies in the world and all the best efforts, we've probably only succeeded properly on about 10% of it, i.e. the trees have grown healthily without any squirrel damage. At the other end of the extreme, we've got complete and utter failure. I mean, write-off. Some of the images that I showed you earlier, that's often not down to us, but often down to owners who haven't done anything, and then you get involved in the estate. Just occasionally it is. Sometimes it's just for the best will in the world, you've got dramatic failure. Sometimes populations can run away with you, and you get overwhelmed and you haven't got enough control measures in place. And then the rest, the 70–80% in the middle, there's a range. On balance, I'd say that's probably 50% we might have got some kind of economic woodland, but there's probably 50% that is utter failure. And I know that there's this dripping tap on the forestry account. I know that there's lost timber money income that is being foregone every year because of past squirrel damage. I think we really need to, because we're just about to splurge on government spending, I think it's really important to take a step back and say, what are these woodlands going to be like in 10 years' time, 20 years' time, if we don't actually sensibly invest in something that could be a game changer and actually could make a decision? I mean, up to this point in time, there's been a challenge over the technology. Trapping is partly effective, shooting is partly effective, IMC might work, warfarin used to work, kind of. This has the potential to do two things, the gene editing work. Not only does it have the potential to keep a complete lid on my squirrels, wherever I happen to be, the great thing about it, it has the potential to control squirrels beyond me, because of the nature of gene spread and gene drives through the population. So then I don't have to keep controlling my squirrels, and it just then starts pushing out. So I think it's a very exciting prospect. We desperately want the support of government to see it through, to say yes, no, we support, but we recognise there's got to be safeguards, there's got to be questions. That's part of what this meeting is about. And at that point, I'll open up the floor.
Graham, thank you very much for that. So if anybody has any questions, we've got about 20 minutes or so for questions. So can I just see if anybody there would like to ask any questions on what you've heard from Bruce and Graham?
Yes, I've got bags of questions.
Okay, Sarah, well, you set the ball rolling.
Thank you. I just worked out how you raise your hand. I mean, thanks very much, both of you, for those fascinating presentations. I mean, can you tell us how this would work in practice? Would you get sort of half a dozen squirrels into captivity, or 20 squirrels, and then when they're in captivity, you would insert the gene drive into them and then let them out, and would you do that in Scotland, and would they then run over the border into England? It sounds a bit naive questions, but what would it really be like when you're not just doing it on Drosophila?
Bruce, you're on mute.
I might not directly address the Scotland question to start with, but happy to touch on that. So at a practical level, you would generate, and I realize things need to change for us to do this, you would generate a small breeding colony of gene-drive grey squirrels. You would continually populate that with females that were not gene drive for two reasons. First of all, the gene drive will be causing female infertility, but Beyond that, you want to keep the gene pool within that small population, because it will be a relatively small population dynamic. You would then, at the right time, take, this is assuming we've gone through all the testing and all the trials in the regulatory process, right? You would then take a number of animals, and the actual number we need to model to determine what that would be, and would release them in a given geographical area. And again, how you'd release them, we need to model. Do you release them half a mile apart? Do you release them 100? We'd have to model all that. So that's how it practically be done at that level. You would then follow up with continuous monitoring of the population there. We should be able to do that through squirrel droppings and just looking at the DNA that's in the droppings, but we may have to have some sentinel animals as well. And from that, you would be able to correlate with your predicted spread of the gene drive, what actually was happening in a given population. And you can also do that further and further away from your release site to actually determine the spread of it. That's how you do it practically. So we'd need to have a small breeding population of greys that were gene drive. Whether you do in Scotland and expect them to march south or north, I think would depend on a variety of things, not least government positioning. But again, we are well away from how we would do trials. The initial trials would be contained in some form or another. I'm not actually anticipating we would set up that colony here in Edinburgh. We could. I think there's more wisdom and an opportunity of setting that up in York with the APHA facility they've got there. And Graham and I have started a dialogue with Giovanna about that type of thinking. But Graham, I know you got thoughts about islands and so forth.
Yeah, all I'd say was, to my mind, I mean, I'm a simple forester, but I understand the concerns, as it were. We don't want a repeat of something going wrong. It must work and must work safely, and there be safeguards for it as it's developed. So, I mean, the key point there, which Bruce didn't labour much, but the daisy chain that he mentioned is one of those safeguards, actually, because you can set the number of cycles that the gene drive works for. So we could set it for 2 or for 5 or for 100, or set it not at all. I think it's important that that's set. The debate is how many times, because you can put a small number with a certain number of cycles, it would have a certain amount of effect, and then it would stop working, and then normal biological behavior would happen, and life carries on as it is. My ideal, I think, if once we've got past the proving the technology works, having a captive bred population, the ideal really, if you want to try it in the wild, would be to have an island, a small island that has grey squirrels on, a wild population, and see how it works there before releasing it into the kind of rest of the UK, as it were. That would be my view, but I'm not the regulator or the or the designer of the scheme. But that's just my view of it.
Thanks, Graham. We've had a question from James Pendlebury. James, you wanted, I just picked up Harvey. Do you want to unmute and to ask the question?
Yeah, happy to, Andrew. I just asked Bruce if he could give us, because I don't know what it currently is, but give us an overview of the current regulatory environment in respect to the possible release of gene-edited animals into the wild, and how that might vary across the country and the constraints and issues that have to be overcome.
I think that's a timely question, but as you're probably aware, DEFRA have just closed a consultation on GM genome editing technology, and we're all waiting with bated breath to hear the outcome of that, which I hope is soon, and I hope is positive towards livestock, of course. But this technology be governed under, at the moment, it'd be governed under GMO technology, and very likely would be governed under GMO technology going forward, since it's a transgene. It does utilize genome editors as the tool, the functional tool, but it is a transgene. Beyond that, there are differences, of course, between the nations around the UK. as there are differences in the density of grey and red squirrels. I think having predictions about how we would unplay that, I'll play Graham's card here and say I'm just a humble scientist. I leave it up to regulators and government to make those decisions for us.
Is it fair to say in Scotland it's not likely to get much traction at the moment?
Absolutely. No, as you know, we're a GM-free country here at the minute. Let me touch on a different way of, as I said, it's up to the regulators and the government to make decisions, and that is their jobs, and some of you are involved in that, and that's great. You may be aware that this morning, Nuffield Council, I'm on the Nuffield Council's working group for genome editing in farm animals, which we hope to report later this year, complete our report. But in advance of that, as part of that overall inquiry, we were involved in our Nuffield Council commissioned a public, what's the right word, exploring public opinion about the technology, and this was released this morning. In that, it's clear that the public see the huge potential of these tools to addressing some of the challenges we have. They have reservations in some areas, but the overriding message was that these tools, if they can help us improve the welfare and can help us to create sustainable food production, this is agricultural use, food production, then these technologies might well have a place in our future. And when we're talking about a gene drive to create sustainable forests, optimize the productivity of forests over a long period of time, forests, as Graham indicated, they're more than just pieces of wood, they're carbon sequestered, they're part of the rural environment, etc., etc. Along with the fact that this is a humane strategy which doesn't involve killing any animal, I think it plays to that public expectation. So governments do make decisions, but they make them on the back of what public want. And Scotland is GM-free at the moment, and it may stay that way. But currently the majority of damage to the grey squirrels cause is south of the border.
Bruce, thank you. We've got a few people are waiting to ask questions. So can we just move on? Martin, I think you want to say something about the regulatory position. You're on mute, Martin.
Sorry. Hi, everybody. Yeah. Thanks, Andrew. I just thought it'd be useful to say a few words. I work for DEFRA on the regulation of GMOs. So just to say that from the presentation that Bruce gave, and I totally agree with him, that these squirrels would be regulated as genetically modified organisms, so the consultation, the outcome of the consultation wouldn't really affect the way the squirrels would be regulated, since they would remain to be GMOs rather than gene-edited organisms, or organisms which could have been produced through traditional methods. Although I should say part of the consultation is the purpose of it is to look at the longer term reform of the GM framework, but that's a much longer term project. So just to say they would be regulated as GMOs, and to say something a bit about the principle of those regulations. And generally we sort of look, the overriding principles are things like GMO would be released in a stepwise fashion, so the scale of the release increases and risk management decreases as knowledge about the GMO and its interaction with the environment increases. And also to say that all decisions on the regulation of these would be made on a case-by-case basis. The other thing to say is probably something about the contained use aspects, because that will be the first step in moving towards a release program. And I think the HSE would be really interested to hear when the technology is ready for moving into this development of the small breeding colony that you mentioned, Bruce, because there are some guidelines on the HSE's website as to the contained use of genetically modified animals, and it might be helpful, to have a look at those guidelines. And the HSE do undertake an inspection program of facilities that are working on so-called larger genetically modified organisms as opposed to genetically modified microorganisms. They have a slightly different set of containment measures and so on. So I think that's hopefully
that's helpful, Martin. Thank you. Richard wants to ask a question, and Mike, but Olivia raised a point a moment ago, which I think was a similar question to the one we had earlier on, Bruce. Is actually, how does this work? How do you make sure that it keeps on going? I think that's what perhaps you could just deal with that quickly, Bruce, and then we can go back to Richard and Mike.
I don't quite understand the question. How we keep it maintained going?
Olivia, do you want to ask the question?
Yeah, I can kind of see that it does work, but it's just the fact that if you're making your squirrels infertile, how do they pass anything on?
All right. Okay. Yes. So
I'm just struggling.
Yeah. And quite rightly too. And I apologize if I didn't get it across to you. That's my failing. So the strategy would be to introduce it into founder male animals, and those males then transmit the gene drive when they reproduce. The offspring from that mating would be male fertile, female infertile, and so on through every generation. So you slowly increase, you don't affect the numbers of boys and girls, but you do reduce the number of fertile females that are around. So you cannot maintain it if it's going to. You're absolutely right. You cannot maintain it if they're infertile. But in the modeling the strategy we're designing, the female is infertile, the male is fertile.
That help. Richard, sorry, Olivia, did you want to ask me?
No, just saying thanks.
Okay, thank you. Richard.
Thank you. Whoever's best placed to answer this, I'm not sure, but what would the risks be that need mitigating here?
What would be the risks? Graham can comment. I'll start off by saying, well, first of all, there isn't a risk from the point of view that the gene drive would transmit to another species. It would be specific, and it could even be specific to subgroups of squirrels. It wouldn't be a risk if a pine marten came along and ate it. The pine marten would have just as good a meal as it did for a non-edited animal. Obviously, there is malicious use of such animals. The animals could be taken somewhere where you don't want them to be taken to, and there's nothing genetic that we can do about that. That's legislative rules that we'd have to put in place to minimize that. There is the risk that and if we look at the homing gene strategy, if we target these infertility genes, there's a risk that the genes have a severe phenotype attached to them that is more than just infertility. And we can learn from mouse studies, but until we actually make an animal, a squirrel, you wouldn't know that. There's a risk that mutations could come in to inactivate your gene drive. Now, those latter two, we can cover by having a series of gene drives ready to go. And the analogy that I would throw at you is the vaccines that we're currently having somebody stick into our arms. If a variant COVID SARS-CoV-2 comes out that escapes the vaccine, the companies have got a series of templates that they can just quickly start to producing alternative vaccines. And and the 12-month wait we had for the first vaccine would probably be cut down to about 3 or 4 months. We can do the same for gene drive. We can have a series of variants that could be deployed to mitigate some of those problems.
I'm glad you mentioned COVID there, Bruce, because of course, you know, I'm not quite sure out of the 12 that have been developed across the world, what proportion are gene edit or have been derived from gene editing, but a good proportion have. And I think going back to the mood question, the public opinion question, I think the mood has changed. It's interesting that that Scotland is a GMO country, but they've all been accepting COVID vaccines quite happily. And some of you may not have picked up that the European Union, which has been completely anti-GMO, last July passed the regulations to allow the vaccines to be developed because of COVID. So I think there's a threshold's been slightly crossed. Going back to the risks, Richard, good question. I just endorse what Bruce says. The biggest concern I have are two fronts. Are what happens if a group of animals gets taken to the native population in America? Part of me would just say, well, welcome to our world, but no, that shouldn't happen. And actually, I doubt it would happen. Regulatory processes would be in place, and also the key thing about the daisy chain being in place is a really good break in the system. You know, if we set a limitless daisy chain, that would be, well, I don't think regulators would be happy, and I don't think we would advise as a sensible route either. But I would just, on the risk front, I would just go reverse the conversation to where I left you. The risk of doing nothing is that we carry on with the same. And that the same is not only having a financial impact, it's having a very big ecological footprint. So it isn't just, and climate change footprint and embarrassment factor in 10 years' time when the government auditors say, well, you spent £700 million and all those trees have been damaged by squirrels. You know, we're talking much bigger amounts of government spending. So I think there's a risk of doing nothing as much as there is a risk of the thing, which clearly we want to bottom out.
Sorry, Mike, you've got a question.
Yes, thanks, Andrew, and thanks to both speakers. I was very interested to hear that this kind of technology had been used in Australia. I think you said on mice, and that it had been endorsed by PETA. I just wonder if you're aware of any kind of social acceptability studies around the use of this technology, and I think I'd be a little reluctant to try and extrapolate that to a different context, but it would still be interesting to see those if they exist.
I'm not aware of specific ones around gene drive, but there will have been some done with respect to the closest probably get to it is around the genome edited mosquitoes that are being released, where these are being released into the wild. They're a wild animal. The studies that have social public acceptance, public opinion, social studies have all been around using these technologies within farmed animals for food production. And I would echo that the situation is somewhat different from where it was 20 years ago with some of the issues remain, and there are some very loud voices, but there's also this growing number, a percentage of our population who see the potential and want to see it being used for the right purpose rather than saying it's just a bad thing to do in the first place. But I'm not aware of specifics about gene drive. And this is one of the reasons why we're speaking to you. Gene drive is really new. If beyond the work that's happening in the US and in labs and this new Australian work and our plan, nothing else is happening in mammals.
Mike, can I just add to that? I think University of Exeter are carrying out a bit of a PR stroke consultancy bit of research on public opinion on this specific thing. There's a meeting next week.
Thanks, Graham. Yes, I've been invited to that.
Ah, right. Okay, fine. Yeah.
Thank you.
So Mike, I was invited to that, but I'm on holiday next week, so family route too far.
Hugh, you've got a question. I'm conscious that It's gone 3 o'clock. If you're happy, we'll go for another sort of 10 minutes. Hugh, your question.
Hi, yeah, thanks. Yeah, my question was about how the daisy chaining actually works. Is that reliant on just kind of the base calculated sort of mutation rate that will break the targeting of it, or how?
No, I'll explain that to you. So it's conceptually, I'll describe it to you. So you're absolutely right. There's a likelihood of mutation coming in here. And it's difficult to know if that's going to be evolutionary scale or if it's going to be more accelerated than that. If it's evolutionary scale, it's not something we really need to worry about, and we wouldn't rely on that as a sort of inactivation. The daisy chain is conceptually, the easiest way of seeing it is a series of, let's say, 3 drives right next door to each other. And drive 1 recognizes 2. So in the first generation, drive 1 goes into 2. In the second generation, 2 goes into 3. In the second generation, 3's got nowhere to go and stops.
Okay.
Because what you're talking about here is a copying of the transgene bit into the target sequence. And if there's no target sequence for it to copy into, it stops.
Yeah. Okay. No, yeah, okay, I understand it now. So the first time it copies in the target sites for the future generations, and then
Yes.
it actually targets itself instead of targeting a gene in the
And then it falls off. It falls off. So you'd engineer that in, well, you could engineer that into the single target site. So it's always at the same location, but the guides that are being used for the next generation only hits the downstream gene drive, and eventually there's no targets for the guides to recognize.
Yeah, I get it. Thanks.
Sarah, you've got your hand is up.
Thank you. Yes, I mean, just to ask Bruce and Graham, really, what are you looking to government for?
Well, do you want to go? Let me say what I'm seeking from government on behalf of ESI. So one is recognition that this is a problem, could be a problem if nothing does change. Secondly, there would be clearly some of this involves regulatory change or specific applications in terms of regulatory approval. Some help and assistance to working that through, so that the answer would be with these protections in place and these regulatory checks in place and blah 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 blah, that the government would be supportive of it. Third part would be some support with funding, because it is this grey squirrel issue is just going to continue to run until we spend some significant amount of money on it, and I've already explained the outcomes, and we know it's been happening for years and years and years and years, and nothing changes. So and we've got finally something that we think could be say game changing.
So I'll paraphrase that, if I may, say that money, but ESI and Edinburgh are looking to the private sector to try and identify individuals who will provide funds into this, and that we're actively doing that. But it would be wonderful if we had the sort of leverage of DEFRA, the government, to support that. But also, just to echo Graham, we need some legislation beyond genetic engineering. We need legislation change to allow us to actually breed greys for a first thing.
Actually, could I just add to that side of your point of view, ESI? What ESI started when the government stopped funding its work into immunocontraception. And that was the sort of trigger that started ESI. So we started to raise money and picked up the IMC project nearly 20 years ago, as Graham has mentioned. And we've been looking at different methods of control. And we now believe we have come up with something, thanks to Bruce's help, which actually can solve the problem. But it's going to cost a lot of money. And we feel that it's government's responsibility now to pick up this board and actually make this happen, because we have a solution. And really only government, through legislative changes, regulatory changes, and cash, can make this happen. It'll actually save the government, a taxpayer, all of us, a huge amount of money. Next question.
It's not really a question. Just to say, government is still funding the immunocontraceptive work being carried out by APHA, both directly through our core funding and through the facilities. So it's not very much.
No, but that did stop in the early 2000s.
Okay,
Yeah,
but we're putting about 50k a year in. And then the other thing just to say is that you can breed grey squirrels. You need a license for it. I mean, Giovanna and co are breeding them at APHA. That's one of the biological challenges I think you probably referred to earlier. It's difficult to get them to breed, but they are breeding them in the APHA facility at St Hutton.
Sarah, I'll just come back on that. Yeah, no, and it's great and it's welcome. I mean, ESI really supports the work that's going on. I mean, we've taken a step back looking at the IMC. If IMC proves to be working, it's brilliant. Where it will take us to is localized control, creating vacuums, coming back. It also has the potential for, you know, it itself is not without some risk. You could get, whilst you design a trap, or design a feeder perfectly, it targets grey squirrels. Some vandal could come through and spread this stuff over the floor, and then other things are affected by it. So it's not an imperfect thing itself, but specifically, it will only get applied where people are prepared to apply it. You create vacuums. It actually doesn't solve the problem. It's like a sticking plaster in that sense. It's a sticking plaster that helps some people do certain things, but it doesn't actually solve the problem. This, we think, has the potential to be a much more dramatic, much more significant national landscape scale impact on the grey squirrel population that is affecting woodland growers everywhere. And other creatures. Yeah, sorry. Sorry, Joan, yes, you got a question. I think David had his hand up.
David, I think David was in first. David, do you want to ask a question?
Yeah, Bruce. In terms of where these technologies have been used in the sort of natural environment rather than under lab conditions, what kind of levels of efficacy have you seen? I'm just kind of thinking about you know, what levels of confidence do you have that if there was significant investment in this, that it would actually deliver?
So the level that what's been achieved in insects ranges into the sort of high percentage of penetrance of the gene drive. There are a variety of papers out there, and it's never reaches 100%, but it's high. It's in the 80s, and the initial mouse study, which had a challenge in our opinion, a design flaw in it, was less than that, and we believe that the sort of design we've got would overcome that. That has to be proven, obviously. So from the point of view of actually driving through the population, it's a bit like this. It's a bit like this coronavirus vaccine of ours. It protects most of us, but not everybody. Now, how you would address that? Well, you could have repeat releases of the gene drive over time. You could have more than one design of the gene drive, which you would release over time, or indeed at the same time. So there are ways to get around that, and we need to do two things. We need to model the types of scenarios we can envisage, but we also eventually need to try it at some point to see what actually happens there. You would get, going back to what Martin was saying, you would get clear evidence about the efficacy of it in the contained use phase of trialing before you went wild.
Yeah, Joe, and then Joan, are you happy with that, Joan?
Sorry, can I speak?
Yeah, Joan. Yeah.
Okay, I'd just like to make 2 points. We've spent a huge amount of time this afternoon with a speaker that told us about the damage that squirrels do. I think that's a battle won. We know that. Forget wasting time making that point. It's well recognized. And I think we should be putting, or you should be putting far more effort into explaining in a much simpler way how this gene drive works. Because I I looked at I'm a molecular biologist, not a gene drive molecular biologist, but I looked at your paper yesterday and I came away from it still not understanding what you're proposing to do. So I think you have a big job in somehow simplifying your message to explain the technology in a far clearer way so that people have a far better understanding of what you're proposing to do.
No, thank you very much, Joan. That's really useful
Sorry,
feedback.
that sounds very critical, but I think until you do that, you're not giving the people the chance to make a kind of balanced decision.
The critical feedback is always the most helpful. Thank you.
Okay.
I think that's a fair comment, Joan, in terms of the only thing I would say is that there are, what I found over the years with people who become forestry regulators, is that not all understand how significant the problem can be, and not all understand what impacts there are. And I think it's I find it quite helpful to explain. I'm amazed that sometimes some people don't always understand quite what's the big deal with grey squirrels. So apologise. There's probably plenty of people in the room that do realise that it's a big deal. It is a big deal. But yeah, no, you're right. Thank you.
Okay. We've got a question from Richard, and I think we probably, unless any other burning questions, 3:15, we'll call that the last one. But there is a recording of this meeting. If anyone would like a copy of that, or if you have any further questions, just email them through to either Graham, Bruce, or myself. So, Richard.
Thank you. To finish on what I hope is a positive note, I want to say thank you very much for bringing us together for this. And it has significantly added to my knowledge. I'm not going to pretend I'm the expert that gets every aspect of it, but on the face of it, there does appear to be quite a compelling economic and even financial case here. I am really keen, therefore, to continue discussing this with you and with DEFRA colleagues and APHA colleagues to actually develop that case. Also notable, the private as well as the public benefits from this. So yes, I'm sure that any advance on this is something where we would be looking to make any investment in partnership and leverage with the private sector. I look forward to discussing further, perhaps Graham, another time, sort of how your other seminar went today as to how fruitful that was looking. But thank you. I think there's something really significant to build on here, and I'm keen to do that with you.
Great.
Thank you.
That is a very positive way to finish. Thank you, Richard.
Thank you. Yeah, thank you. Thank you, everybody.
Can I just say that I think the modelling of that financial case is the interesting thing, Richard, because it would be quite, I think we can do it. On the feedback I've had from talking privately to personal clients is that they get it. They're the ones who are having to write the dripping tax check every year, whether it's 5, 10, 15 grand a year or more, and they would like to see this happen. They would be supportive of it happening, and probably willing to, if it was guaranteed to work, they'd be happy to write the check to support it as well. So yeah. Anyway, thank you. It's nice to meet you all. Hopefully we'll be able to see each other face to face at some point, because I get fed up of Zoom. Good. So thank you all. Enjoy your rest of your Friday afternoons, and hopefully we'll go and have some more discussions. And I think I'd like to follow up on some of that discussions, Richard, and see where we can go. Okay.
Thank you.
Thank you.
Cheers. Bye bye.
Thanks very much.